Live. It's getting us all set up. I'm going to mute myself so it's not a glaring noise in the YouTube. All right. And we are officially live on YouTube. So for folks who want the YouTube recording after the event, it's in the chat. I'll tweet it out on SciStarter too. And there's Pietro. Hey, Pietro.
Hello.
Robin's going to be emceeing you today and advancing your slides. Just as an FYI, after we do our initial poll questions, it's right over to you because this is the companion event to last week. So we're not going to explain SciStarter or citizen science. We're just going to give a brief primer on stall catchers and then go ahead and ask all those questions. Do you have any questions for us? We already have some attendees in here. So attendees, feel free to say hello in the chat and let us know where you're calling in from. And feel free to, if you have questions for Pietro already, put those in the chat too. But Pietro, do you need anything else before we kind of get started?
Can I get a Starbucks real quick?
Yeah, go for it. We'll get started in 2 minutes.
I'm just kidding. I'm good to go.
Oh, okay.
Yeah. I wish I could do that, but I don't see one nearby. So
there's not one in your house. You don't have your neighborhood Starbucks. Okay.
No, I need to do something about this. Yeah.
For the rest of our organizing committee, do any of you have any last minute questions?
I think I'm all set over here.
All right, thanks, Michael. And yeah, when you get to the 10-minute mark with 10 minutes left in the webinar, we'll switch to you and we'll end the Q&A. So for our attendees who are here, you're here for a very special event. We've never done like an Ask Me Anything style event before. So you're going to get all your questions about stall catchers answered from Dr. Pietro Michelucci, the founder of Stall Catchers. Robin Salthouse is your MC today. So since we're a minute out, I'm going to leave it in her capable hands.
Okay, welcome everybody. Please put your zip code and where you're from in our chat. We'd love to know where you are. Feel free to ask questions and any other thoughts you have on this great program we're presenting. I'm going to start sharing the slides. And so let's see. All right, well, it's start time. Welcome everybody. My name is Robin Salthouse. I'm with the SciStarter organization. And today we're going to follow up on a program we had a week ago where we're going to talk about the results of participating in the Stall Catcher Citizen Science program. This is being supported by NNLM and the All of Us Research Program. So thank you so much for attending. So we're going to start out with some polls. First of all, we'd like to know where you're coming from. And so if you would please put your zip code in the chat, it really helps us to know who's attending these, where you're from, and welcome. So our first poll is, did you attend last week's event? So it's right up there for you to do. Please go ahead and put your response in, and then we'll share the results. See, there you go. So if you'd please participate and let us know if you were at last week's event. So it seems like the majority, if not all of our attendees were at last week's event. That's great. We're excited to find out what happened since last week. So I'm getting ready to end the poll. If you haven't answered yet, please do so now. Okay, 5, 4, 3, 2, 1. Okay, and so everybody who was here last week, or not everybody, but everybody that's here today was at last week's event. Thank you so much for participating in that. So let's go to the next poll. Have you visited or joined the all of Us research project and learned more about NLM as well? So again, if you could let us know, have you visited, joined the allofus.org NLM to learn more about All of Us research? Okay, so again, if you could just finish up that poll, 3, 2, 1, I'm going to end it. And here's the results. So that's great. We really appreciate that you went from this webinar and got more familiar with the All of Us research project. Thank you. Okay, so let's go to the next poll question. Again, we'd like to know who you are. Are you an aspiring citizen scientist? Are you a parent or an aspiring citizen scientist, a teacher, an educator? Are you a librarian, a researcher, or project leader, or just a bored person on the internet who's happened to join us today? And if you're none of these, that's okay too. We want to know about that. Okay, so it looks like everybody has participated, and I'm going to end the poll. And here's the results. So that's wonderful. We have a lot of aspiring citizen scientists. And we even have somebody that was bored and joined us from the internet. That's great. And I'm glad to have those parents and librarians too, because you're helping educate your community or your children on citizen science. Thank you so much. Okay, so let's continue on with our presentation. Okay, Pietro, I'm going to turn it over to you now.
Okay. Hello, everyone. Did we want to go back over a bit about this background on the project? I see the slide in front of me before we continue. In order to repeat some of what we covered last week, or do we want to just launch right into some Q&A?
Just a quick just a very, brief
Sure.
explanation. Yeah, thank you.
Okay, sure. Yep. So the basic idea here is that the Cornell biomedical department has a lab called the Schaefer Nishimura lab, where they made an important discovery about the role of brain blood flow in Alzheimer's disease. So we've known since the beginning of Alzheimer's that patients tend to have about 30% less brain blood flow, which is equivalent to that feeling you get if you've been lying down and you suddenly stand up and you feel a little lightheaded. That's sort of present all the time, that kind of that lack of oxygenation in the brain. So the big question is, what's causing this? What are the underlying mechanisms for this in Alzheimer's disease? And to what extent is that actually related to the cognitive deficits that we observe in the disease? And if we can restore that blood flow, do those cognitive issues go away? So next slide, please. What they discovered in their early research in the lab is that first of all, the underlying mechanism for the reduced brain blood flow is that the tiniest vessels in the brain, called capillaries, were actually stalling more often than they do in healthy people. So everyone gets these occasional stalls in our capillaries, and that's not considered a disease. That's just part of being a human. And they only last a few seconds, and then the blood starts flowing again. And often this is due to an inflammatory process. So when we think of inflammation, we often think of swelling, but when we talk about inflammation in the brain in these capillaries, we're really talking about an immunologic response where a lymphocyte, a white blood cell, which is used to fight infection, gets deployed and finds its way into one of these capillaries, and then there's a little hook in the side of the capillary which activates and hooks onto that white blood cell. That's the actual mechanism that that we refer to as inflammation. And when that happens in a capillary, that blood cell blocks the flow. I mean, these blood cells are so narrow that the red blood cells have to line up one by one in a row just to get through. So as soon as you get anything blocking that, everything just stops. And when you play stall catchers, and maybe you have played it already, then you've probably seen some instances of this where you actually see these dark spots in the vessel that have just stopped. They're not flowing at all. And that's because there's this stuck white blood cell from this inflammation. So, so what they did in the lab is they were actually able to release these stalls and restore blood flow in these mouse models of Alzheimer's disease, mice that have been given the human Alzheimer's gene and manifest all of the physiology of Alzheimer's. And when the blood flow was restored, when the capillaries were not stuck anymore, and they saw a complete restoration of blood flow back to normal levels in the brain, which was evidence supporting the idea that it was the capillaries that were causing this. And they also saw that the mice actually got their memories back, and some of the cognitive symptoms they had from Alzheimer's actually reversed or completely resolved. What's amazing is that this resolution of cognitive symptoms happened on a time scale of only a few hours. So that's how quickly these symptoms went away. So this was very exciting. The problem is that the way they released these stalls in the mouse brains required an immunosuppressant that would not be sustainable. It would make a mouse or a human, if it became a human treatment, prone to infection. It would be like giving them AIDS. So in this case, the cure would be worse than the disease. So then the goal became to find a treatment that could reduce these stalls without having these negative side effects. And that's the work of stall catchers today. Next slide, please. So I think I mentioned that when they were able to restore the blood flow, the symptoms in the mice went away. The problem was that the mechanism for restoring the blood flow created new problems. Next slide. And next slide again. So, so the reason we created stall catchers is because the issue they had at the lab was that each experiment required them to introduce a potential treatment, for example, to mice that were given the Alzheimer's disease, and see whether or not it was actually reducing the stalls and what other kinds of effects they had on the mice, and this. The way that they conduct these experiments is once the mice have been given the treatment, then they're imaged under a digital microscope, and that produces these what we call image stacks. They're like volumetric images. Imagine sort of, instead of a flat image, a whole cube of image. And when you're scrolling, when you play stall catchers, that scrolling back and forth is actually looking down through layers of that image stack. So you're actually moving through this volumetric image. And the problem they had is that to go and look at every individual capillary and try to figure out whether blood was flowing through it or not would take them 6 months to a year just for one experiment, which meant that all the experiments that they anticipated needing to do to come to a treatment for Alzheimer's based on this approach could take as long as 30 years. And obviously we want to get to a treatment as soon as we can, and hopefully much sooner than 30 years from now. So our idea was, if we could take this task that they were doing in the laboratory, and here you can see a laboratory technician doing kind of a version of stall catchers the way they used to do it in the lab. If we could isolate each capillary using automated methods, draw an outline around it, and then show that same capillary to several different people and get a kind of a consensus voting of whether it's flowing or stalled, we could potentially convert that into an expert-like answer and do this much more rapidly by getting hundreds or even thousands of people engaging in this task online at the same time. So that was kind of the vision behind stall catchers, and today it's the reality of stall catchers, and we validated this approach. Next slide, please. And it's we're analyzing the data at least 5 times. Last time we evaluated, we were analyzing 5 times faster than the lab. I think it could be even closer to 10 times as fast as the lab now. In fact, the lab is now struggling to give us the data as fast as we've been analyzing it. So, and that's something actually we have an idea for how to enlist help from stall catchers players to make that process even faster for the lab as well. So I think we can potentially speed this up even more than we ever expected to. Next. Yep, so this should look familiar if you've been playing stall catchers. This is what it looks like on a tablet, and you can see there's an orange outline around a capillary. You can't, in this image, you can't really see the capillary. You'd have to scroll that little green slider button over, and then you would see it appear inside the outline. And then you'd make a judgment about whether blood looked like it was flowing in a particular direction or if it was just kind of stuck. Next slide. Yep, so I think I mentioned this. We actually need to update these statistics. We have about 35,000 people playing stall catchers now. On any given day, probably 50 to 150 people. Sometimes when we have events, we could see 1,000 people, different people playing in a given day. Yeah, and I think we really are analyzing data even faster than what's represented here. Next, please. Okay, so that I guess brings us back to the Q&A, and I thought what I might do is just begin. There was a question last week that I didn't have the answer to, and I think it was a great question. So I went to the lab and I re-asked this question, which they've probably answered for me 2 or 3 times by now. The question was something like, which part of the brain is being imaged? Which part of the mouse brain are we looking at when we play stall catchers? So I thought I would just start by answering that. So it turns out it is part of the prefrontal cortex, which is the most evolved part of the brain. It's usually, I think, what we think of when we imagine, what a brain looks like. We're thinking of all these convoluted lobes. That's the prefrontal cortex. And in this case, it's the parietal lobe, which is involved in processing sensory inputs, both at the surface of the body and inside the body, like interoception. You can actually, feel things in your body. And I think there are regions also involved in motor control that are represented in these images. So it's about sensing and acting, those parts of the brain. So I hope that is helpful to answering that question, and happy to discuss or answer any other questions.
Well, thank you, Pietro. And we did grab some of the questions, which you've already answered one. I guess maybe should we start off with how did our team do this week? Caroline created a team last week, and were you able to monitor our results, or do you have any comments on any outstanding team participation as far as the event or any of the groups went last week? Because I know you have some real stars.
Yes. So I'm thinking if we can bring up stall catchers here. Do you want to do it, or do you want me to share my screen and bring it up? I can do it either way.
Do you mind bringing it up?
Not at all.
Thank you.
Bear with me one moment, please, while I get that set up.
Sure.
Yeah, it looks like we did have some nice contributions here to the research. Some folks really spent some time on this, I can see. All right, I'm going to go ahead and share.
Whoops.
Oh.
Oh, do you want, yeah,
I think you have to let go of sharing your screen.
I will stop and let you share. Thank you.
Okay, great. And then, yeah, let's see if I can.
And then I'm going to put the team connection in our chat too.
Okay, great. Can you see my screen here now?
Yes.
Okay, excellent. So, so it looks like we had 19 participants on this team. This is the team that we created last week. A team score of 683,000 points, which is pretty good for a brand new team, because it's slower to get points when you're less experienced. So that's quite impressive to me. The overall team rank, 123. So, well, that's probably in the history of time. So that makes sense. But I think the key thing here is to look at the amount of research that we accomplished. So it looks like, so I think I explained last week that one of the ways that we're able to properly combine many different answers about the same vessel from different people is we sort of track how well people are doing in the game and figure out who has increased sensitivity for certain kinds of vessels. So then when you have this kind of voting situation, so you know, Robin, you might say flowing, I might say stalled, Caroline might say flowing, and then we need to kind of assign a weight to each one of our answers. It's not just a simple vote. It could be, Robin, since you've been playing for a long time and you're much more experienced, that your vote counts much more than mine does. So in order to figure that out, we have these, we show vessels where we already know the answer, and then we see how people do, and that lets us sort of track over time how they're doing and assign this weight. So that's what these training movies are down here. As people become more experienced, they see fewer and fewer training movies and more of the research movies. The research vessel movies are the ones where we're actually getting answers for the first time that are directly contributing to the Cornell research. So overall, it looks like about 1,300 vessels were analyzed over the last week by this group. That's a lot of work. 1,300 vessels. That is not nothing. And in terms of research vessels, we analyzed 537. So I'm going to do a little, I'm going to cheat and use my calculator here to figure out the lab equivalent. So that's
great.
Yeah, that's fantastic. So that's the equivalent of 16 hours worth of lab time, almost 17 hours worth of lab time. So, you know, that's a real research contribution. That's a couple days of laboratory analysis that does not have to happen now because of this contribution. So right. I'm really proud of this work, and I'm really grateful to everyone who contributed to this. I see we have some Polly Lover as the username here was the top contributor. It looks like the top three were in somewhat close contention. Polly Lover, NS Kate, and Sunshine 2020 were some heavy lifters here, and and all contributed as recently as today, and many others here. Every single person contributed to this research. So who's on this list. So thank you very much for that. Yeah, that's a great contribution.
Yeah, it is. And we have some questions. So somebody is asking, there seems to be different levels of magnification in different samples. Are the RBCs the same size in all the video samples?
Yeah, that's a really good question. I think this is one of those sort of theory and practice things where in theory they're all supposed to be the same. In practice, I'm not sure how consistent they actually are. If you're seeing huge differences, I don't think that's right. I think so. Sometimes we get images that include the bigger vessels like arterioles or venules, and these are mistakenly included and outlined, and in those cases, they're all going to be flowing. If they're not flowing, it suggests there's like an actual stroke going on. So in almost all cases, these will be flowing and shouldn't be included among the capillaries. So if you see a really fat vessel, that's probably not a capillary and is just included accidentally. So we don't have a perfect preprocessor. If we did, none of those would be showing up.
Well, and a follow-up question is, can you see stuck WBCs?
I think we see them. I don't think they show up explicitly as the dark spot. As far as I understand it, they're creating a blockage, and then it's the red blood cells that are actually the dark spots that are lined up, stuck behind the white blood cell. But I'll double. That's a good question. I've wondered about that myself. So I'm gonna go back to the team and ask about that.
Okay, great, thank you. And I like this question too. How are the orange areas chosen?
So that's a really tricky part. We spent a lot of time working on that. So it's done by a pretty fancy computer algorithm that goes through and tries to figure out for each pixel in an image stack, does that pixel represent vessel tissue or not vessel tissue? And if it does represent vessel tissue, then what are the blood vessel, where are all the blood vessel segments? So blood vessel segment is a blood vessel that connects at each end to other blood vessels. So so the first part of the challenge is figuring out, which parts are the vessels themselves, and where we do see vessels, where do they appear to be embedded between two junctions, and then how do we carefully draw an outline around that? And we use a completely automated process today to draw these outlines. So that's very fast, but it's also not completely error-free, as you've probably seen. Sometimes you might see an outline that doesn't include a vessel. That's an error. Or you might see an outline that includes one of these, fat vessels that shouldn't be there. And so we do have a little flag. If you see something that doesn't look quite right, you can still answer. In fact, you have to answer flowing or stalled to move on to the next one. But you can also click on the flag before you answer, and that just lets us know, there could be a problem with this outline or this vessel. And then the researchers actually go back and carefully look at those, and will exclude them if they shouldn't be there.
Well, and so that's kind of, I think, helps answer this next follow-up question. Some films and the selected areas seem to show nothing but static. How should we mark these? So is this where you would use that click on the flag?
Yeah, that's exactly what I would do. Especially if it's just a whole field of static, I call it snow, then yeah, that certainly doesn't sound right. There are occasionally, sometimes we get very noisy images where to someone who's very experienced with stall catchers, they might still be able to tease out, the appearance of a vessel and make some judgment about it. But yeah, I would say just flag those.
Okay. And, PHO, especially for people who are librarians or parents homeschooling or just doing the whole pandemic education thing right now, do stall catchers offer classroom resources or, other help for, people who want to go a little bit more into the whole stall catcher program?
Yeah, we do have some resources that are available. So I'm on the Stall Catchers website here still. I'm going to go to the About menu and click on Science. We have an explanation of the underlying science. We have some videos as well. Let's see. Yep. So let's see if we go back up to About. For educators, we have a section under How To, and you can see there's this menu here of downloadable materials, stall catchers in schools, and if you go to the we have some very talented people on our team who have developed some really solid science communication videos. They tend to be brief, kind of manageable amounts, even by today's limited attention span standards. So I think this video is maybe 2 or 3 minutes long, and it just explains how playing stall catchers actually helps advance the research.
Yeah. That's really great because, in past webinars we've done with you, we've had several teachers on. And so I think it's really nice to have these kind of resources to go back to. And also these webinars are always posted and archived on our YouTube, SciStarter YouTube account, as well as the SciStarter/NLM account. In fact, this one will be too. So, don't forget to come back to these archived recordings and, find out about this information. So thank you so much. All right. And then I guess we've talked about this before, but how have libraries been part of stall catchers?
Yeah, so libraries, it seems like today are serving for many communities as community centers, and certainly places where people can come together not just to find textual resources, but to engage in community activities. And we saw a real opportunity with NNLM to partner on a big event we called the Megathon. It was our most ambitious public event where our goal was to analyze an entire year's worth of research data, analyze an entire experiment in just a single event. And after, I think what ended up being a long weekend, we had a few technical issues. So it eked into Monday. It was started Friday, Saturday, Sunday, and then into Monday. And we actually did analyze an entire data set. We did that in partnership with the library. So each librarian at the different libraries participated in outreach to the local communities to get folks involved. And it was just so exciting to be able to say, look at this, when we were at the very beginning of stall catchers, it would take the lab 6 months to a year to answer a research question. And we just did it together as a country, as a bunch of local communities coming together at their libraries or from home connected to their libraries. And we answered a research question in a weekend.
Yeah, and I was part of that megathon with the library I worked at, and it's so amazing when people have a way to contribute. We had a retired nurse on our team who really delved into this, and I'm hoping I'm going to answer the next question somebody had about can more experienced people say whether it gets easier and or faster. And I think what they're asking is, as you become more and more involved in this project and help contribute to it. So I know the retired nurse that was on our library team just really spent some time on it. She made a lot of contributions, and we actually looked at her to help us kind of do a better job. And I think the answer to that question is yes. With the time she spent on it, she certainly helped me kind of talk about, well, this is why this is stalled, or this is why this film is flowing. So my own personal experience is the more you contribute, the faster and easier things go. You kind of learn the lay of the land. And I guess, would you agree with that answer, Pietro?
Yeah, Robin, I would agree completely. So what we've seen is that for anyone coming to stall catchers for the first time, there are some people who just have an instant knack for it. They somehow, and I'm not one of them, by the way, but some people can just come to it and they get it, they see it, they somehow do it very easily at first, or certainly more easily than others. There are others who kind of they get them even backwards at first. They think the flowing ones are stalled and the stalled ones are flowing. Well, actually, even if you've got them backwards, if you can tell them apart, you're actually halfway there. You just have to kind of switch your labels. We saw this at the American Science and Technology Fair. When we started to, the project had just launched, and we had an exhibit there with SciStarter for stall catchers, and families would come by with young children and their parents, and at first it looked like, oh, the parents get it, the children are kind of struggling with it, but then the next family would come along, the children would get it easily and the parents wouldn't understand it. So it's not really an age-related thing as far as we can tell. It's really just person to person. But the universal pattern that we notice, and I think this is what you're getting at, is that regardless of where anyone starts, the longer they play, the easier it gets. I mean, we have all kinds of, I could show you graphs. We have trends. Everybody gets better. It gets easier for everybody over time. You just have to get used to it, basically. And for me, so I think this is flowing. I'm going to guess flowing on this one if you're still watching. Okay, it's a research vessel. We don't know yet. I was hoping to find one where there were some comments at the bottom. Let's see.
Yeah, and this goes to another question I have is, your community. And that's what's so amazing, is that you have built a community around this project, and people are providing comments, or they, get involved in kind of your online posts. It's really kind of neat.
I'm going to tell them, hey, everyone, we are on a live SciStarter NNLM webinar, hanging out here with you today.
Yeah.
There we go.
Yeah, what a cool way to engage with your fellow catchers. And somebody also has just recently asked in our Q&A, is there a way to magnify small areas within the video?
That's been a feature request for us. And there are a lot of considerations, I think, that go into something like that. I do know that there are folks who will use their browser magnification buttons to just kind of make everything look bigger. So there are some workarounds to that. We have very early in the project, we had the idea of zooming into the size of the outline, so you could get as close as possible and still see the entire vessel. The issue there is that would create a scaling issue related to what one of the earlier questions was about, where you might feel like you're comparing apples to oranges, because sometimes things would look big and then you couldn't tell if that was the same thing or comparable to what you saw in the previous video. So for now, we've sort of standardized on a single size, but I think it's fine to play around with the browser magnification. For a lot of browsers and computers, it's something like Command plus or Control plus, that key combination. And I think there's yeah, there's even a menu here. I don't know if you can see way up here.
Yeah,
There's an option on Chrome to zoom in or zoom out.
Yeah.
Yeah.
Yes. Yeah, we can
So,
see that. That's
yep.
great. Well, that's a great tip, but it also makes sense that, we gotta, like you say, compare apples to apples. And, so how is Stall Catchers going to be involved with Citizen Science Month? How is that looking for you? And is this another way the viewers can get involved, or maybe even pull some more friends or family in to get involved in this project? Is in April during Citizen Science Month.
Yeah, great question. So I don't know if Caroline is still on the line with us, but someone asked last time if we were going to be running an event anytime soon, and so I invited Caroline to organize one with me in response to that request, because we have been wanting to do one, and I think Citizen Science Month would be a great opportunity to do that. So if I remember right, I think that's when we're aiming to do an event, and of course we'll make all the information broadly available as we get closer to that. But I think it would be a lot of fun.
Yeah, and she put in chat, she's ready. She's going to be working with you to create that event. And I just put
Fantastic.
into chat, yeah. And I mean, it's so amazing the results that come from those kind of events. And I would encourage everybody to go to scistarter.org/events to keep an eye on that event coming up. And usually Caroline's on top of things, and that'll be posted well in advance so you can start practicing and get involved if necessary, form teams. So yeah, keep an eye on that. Also, don't forget, as Caroline is reminding us, to go to citizensciencemonth.org, especially if you're a librarian or a community group who wants to get involved and work with your community to get people involved in this citizen science project or others that you may already be working on. So don't forget to check out all the great resources on the webpage Citizen Science Month, all one word, .org. So what is the biggest change, Pietro, you've made to Stall Catchers?
The biggest change, you mean in recently,
Well,
as far as the
yeah, probably the web. Yes, the website, because I know in past discussions we've had, you've talked about how, much the community has provided updates. So, you explained why you couldn't do the magnification, but I know you've made changes because of the community's suggestions. And have there been any recent ones, whether they were community or something you all felt that, would help the project?
Yeah, no, that's a great question. So we have had tremendous input from the community over the years. I think I mentioned we visited Boise, Hazelwood Middle School in Boise, Idaho. I spent a day with the students there, and they gave me a laundry list of features. We still haven't added avatars and given options for dressing them up. That's actually something we'd love to do. I think,
Yeah.
We'd reach a lot of students with that. Most recently, we had some performance issues come up. So one of our most prolific contributors was noticed that others were able to answer movies, to load movies and respond to them 2 or 3 times as fast as he could. And this was frustrating because he's a very sensitive player. I mean, he's a very skilled player, and he can answer much more rapidly than the interface was allowing him to answer. So we looked into it, and what we discovered is that so one of the challenges here is to figure out when we're showing someone a movie whether or not you've seen this movie before. So Robin, we don't want to show you the same movie twice. We don't want to get the two answers from you about the same movie, because that would kind of be like letting you vote twice, right? Which wouldn't be fair.
Right.
So
Sure.
we always have to go back, and every time we're looking for a new movie to give you, we have to look through all the movies that you've already seen before. Well, it turns out that for this prolific player, there were so many movies to look through in order to pick the next movie that it was taking longer.
Yeah, that makes total sense. I mean, in a way, it's a good problem, but wow,
Right.
how frustrating for him, because he's in there and wants to contribute.
Exactly. So fortunately, we figured out a pretty simple workaround. What we realized is that even though in theory we could have multiple data sets active in stall catchers at the same time, we've never actually used it that way. So there was no need to look through all the old data sets that he had analyzed, only the current data set. So we just switched it to only look at the current data set, and then it was just, I think he described it as a night and day difference in performance. So he is now up and running at top speed again. And anyone else who's a prolific catcher would enjoy those same benefits. And in fact, we heard from a few others who said, wow, system suddenly working a lot smoother. So
That
that's great.
that's great. Yeah. And again, I think that's kudos to your organization, taking those comments and those suggestions and only making your project better and better. And on that note, maybe you can speak to how you even include these contributors into your research papers, into your publications.
Yeah, you bet. That's something we really enjoy doing. So basically, the idea is that if you're a researcher, a scientist, and you're publishing a research paper, then there's a set of ethics and guidelines about who should be included as an author on the paper, and there are even rules about the order of the authors in terms of the kinds of contributions they make. In academia, this can become something that people really pay a lot of attention to, and we felt like this is a very specific kind of research contribution that merits some level of co-authorship and should be recognized in some way in the actual research publications. So, and we're not the first citizen science project to do this. We saw some precedents from other projects that we hold in high esteem. The Stardust at Home project has been a real model for us. And what they did is they took a list of all of their volunteer contributors and made a website with just that list of usernames. And then in the actual scientific publication, they mentioned their contributors and then included a link on the author line. They said, the Stardust at Home players, and then they included a link to that list. So you could go and see the usernames of every single person who contributed to that research. And we do it as much as we're allowed to. Different journals have different publication policies for this, but we're really trying to push the envelope and also get the publication industry, the scientific publication industry, to kind of shift to this view a bit more. And one of the things we do is we don't just list everyone who's ever contributed to stall catchers. We actually know for each data set who contributed to that data set. So if you want to be listed as an author in a publication for each data set, just show up once a month and make sure you just look at one vessel for each data set, and then you'll get all kinds of publication credits for doing that. We also in our listings, I don't know if other projects do this, but we list usernames in order of contribution. So the person who contributed the most gets listed first, and that's kind of consistent with how authorship works in the science world.
Yeah, that makes sense. And what a great illustration that citizen science can be done by anyone, that you're making important contributions by crowdsourcing data contributions. So that's really great. You're doing this for your community. I've got a couple more questions from Q&A. One is, can you review slides where there is more than one vein and what to look for? But don't most slides only, they just allow you to focus on one veinet or one capillary at a time, correct?
Well, ideally, but in practice, things are messy. This is biology, and the brains of these mice did not evolve to fit perfectly into our outlines.
Sure.
So here's an example. I'm looking at one now. If you start looking at the movie over on this part of the slider, you can see there's this vessel up here, right? I don't know if you can see my mouse cursor.
Oh yeah, no,
it's a mouse cursor on a mouse brain. And little stall catcher's humor there. But now if you move the slider over to the left, then you suddenly see there's this other vessel here that's actually following the shape of this outline more centrally. And that's always our kind of rule of thumb, is if there are multiple vessels inside an outline, you don't want the one that's going across the outline or diagonal, the one that's really fitting as closely as possible to the shape of the outline. And, I wouldn't worry too much about it. If you get a very ambiguous situation, make your best guess and don't deliberate too much about it. Just make your best guess and then move on to the next one. In the end, if there's a problem with a vessel, if there are two vessels and they look equally likely, that'll kind of get sorted out just from the numbers in the end. It's not likely to harm the research in any way.
Great. Well, that was another good tip for users. And then here is another question, and we're going to probably this will be our last question, and we'll move on with today's program. The question is, the probability seems much greater for flow. If uncertain, do I do flow as default? Is this taken into account statistically?
Yeah, what a wonderful question. I'd be curious to know the background of the person who asked that. Yeah, so yes, the likelihood of a stall is greater. And we actually have programmed that likelihood to remain fairly constant. So people who, and I can just tell you, you're gonna see flowing about 4 times more often than stalled. 4 or 5 times more often than stalled. And we control that intentionally because, people will tend to optimize to what we call the base rates. So these are the base rates. The base rate is kind of the expected rate at which the expected likelihood of seeing a stalled versus a flowing vessel. And if we were varying those base rates, then people would get confused and they'd start kind of adjusting their bias towards saying flowing all the time or stalled all the time. And then that could alter the results. So I think the answer to the question is, if you're unsure, should you say flowing? I would say if you're unsure, just lean in whichever direction you think the vessel is actually going. So don't try to play the base rates. Say what you actually think it is. Sometimes we really just can't tell, and pick at random then. That's what I would say. Pick at random. We actually we track not just sensitivity but bias. So if there are people who tend to say flowing when they're not sure all the time, we know that. We factor that into our analysis, and or I should say we factor that out of the analysis so it's not affecting the ultimate decision about the vessel. So if there are folks who want to delve deeper into our analytic methods, I'd be happy to take that offline, answer an email or share it in other ways. We might even have a video about it.
Okay. Well, Pietro, thank you very much for that. And I'm going to get the slides back up. And in the meantime, I'm going to have Michael come on, who's going to tell us about the All of Us research project.
And hey everyone, this is Michael Balkenhol. Some of you may have heard me last week. I work for the network of the National Library of Medicine, which is a program of the National Library of Medicine. And we are one of the supporters of today's event. So some of our work is working to, with the All of Us research program, to essentially just raise awareness of it. So All of Us is a research program of the National Institute of Health. And they are seeking 1 million or more people from across the US to help speed up medical research. And people who join will sign up to share information about their health habits, what it's like where they live. And by looking for patterns, researchers will learn more about what affects people's health. You can go to the next slide, Robin. So All of Us is a precision medicine study, which is essentially just another way that researchers are looking to advance the ability to serve the public health needs of all people. It's a way of looking at medicine as being more individualized. This way, doctors can take into account a person's lifestyle, environment, and biological makeup to find out the best treatment for the individual. You can hop to the next slide. So the neat thing is that we already use precision medicine in some ways, such as with eyeglass prescriptions. Eye doctors can tell us exactly which lenses work best for us, but when we go visit our regular doctor, they may be more likely to prescribe the treatment or prevention that works well for most people. But not everyone's needs are the same. So All of Us is building a large national database to help researchers eliminate one-size-fits-all medicine. But to do this, they do need volunteers from all walks of life to advance this work. If you want to hop to the next slide, Robin. So if you are curious, this is a new slide from that wasn't here last week. If you would like to learn more about the connection between citizen science and the importance of participating in medical research, check out this SciStarter blog, and I will drop it in the chat in a moment. This was a SciStarter blog post by Eric Betts that shares in detail how All of Us got started and where it is going. You can hop to my next slide, Robin. So lastly, the reason for today, citizen scientists understand the importance of volunteering to advance scientific research. And as I shared, when we're talking about All of Us, the future of individualized medicine may depend on an army of 1 million or more volunteers, and scientists do want you to get involved. So to learn more about All of Us and whether participating is right for you, please do visit joinallofus.org/NLM to learn a little bit about the program. And thanks for listening, everyone. It was really interesting. Thanks, Pietro. I've been playing over the last week, and I think I'm getting better at it, maybe. So it was a pretty good time. So back to you, Robin, to wrap us up for the day.
Sure. Thank you, Michael. And again, thank you, NNLM, for all your generous support of this program series. So in order to keep our programs relevant and the best they could possibly be, we really need your help in feedback. So if you could please help us by filling out this survey, we'd really appreciate it. So here it is. If you would put that in your browser and just take a couple minutes to help us out and give us feedback on this event. We really appreciate it. Let's see, and then we have some more programs coming up, but we'd certainly like you to continue participating in stall catchers, and you can find links to it through the scistarter.org/NLM site. And then we're going to continue this series with another program with a similar format. It's going to be based on the Eterna project, and that will be in February. So please go into scistarter.org/NLM to find the link to register or you can go into scistarter.org/events and look in February for this Eterna program, where we'll learn about the project. We'll, of course, answer your questions. We'll send you off to participate. And then a week later, we'll come back to look at the results and answer any other questions you may have had while participating in the program. And this is all about RNA-based medicines. So, with Eterna, you're helping scientists develop RNA-based medicines by solving puzzles. And, on February 18th, we'll talk about the goals and tasks. And then the following week, February 25th, we'll go ahead and we'll do as we did today. We'll maybe delve into more specific questions you may have regarding the program. So we're wrapping up today. I'd like to thank Pietro again with Stall Catchers. He's been amazing in his support and of citizen science, especially with Stall Catchers. And we have several people with NLM. Thank you so much. I guess I'll ask you guys, do you have any other comments or thoughts from today's program, or anything else you wanted to add for today? Okay, and
I just want to say thanks to everyone for joining the webinar. This has been a very interesting group to engage with. I've engaged with a lot of groups. I thought there were some and I always enjoy it and get different things out of it. In this case, there were some really apt questions, and I really appreciated that engagement. And I hope you'll continue to try Stall Catchers and see if it gets easier. And there are a lot of other great citizen science projects out there too. I think Eterna, the one, if you show up next time, is also a fantastic project. So thanks again.
Okay,
thanks for having me.
Great. And I see somebody said that there was an issue with the survey links, so we'll look into that. Sorry about that, but there will be a follow-up email to everyone with the recorded link of this, and again with the survey. So and here we've put it in again. So here's the survey if that helps you all. Thank you, Michael, for putting that in there. But there will be a follow-up email with these links as well. So again, thank you everyone for joining us, and hopefully we'll see you back in February. Thanks, everyone.
All right. Thanks, Robin. Have a good day, everybody. Ready for me to close down